Okay, thank you all for coming out. I don't want to be too loud for the people in the front or too quiet for the people in the back. So he wants louder in the back. Okay, we can be loud too. All right. So my name is Bob Kinzie. I was at the zoology department at the University of Hawaii. I left the department and then the department disappeared. There was no longer there. I'm not sure if there's a connection or not. Okay, and as you heard, we're going to be talking about jellyfish and how maybe they live forever and what we can learn from that. Okay, so start, because as professors always start with outlines to tell you what's coming. We'll go over some basics of cnidarian biology. I think this will be familiar to a lot of you. And so I'm going to go pretty fast because I want to leave space at the end for questions. If I go too fast, yell and say, I didn't understand that, and I'll try to understand it some more. Okay, then we'll talk about sex. Then we'll talk about why cell division is bad for you. And then get to the main point of the talk, which is some experiments that a student of mine did. And then see what we learned from that. And then there'll be a quiz afterwards, and if you pass the quiz, we'll let you take your car out of the parking lot. Okay, so this is the review part. I think a lot of you know this story, the cnidarians. There are several different groups, the Anthozoa. The Anthozoa are jellyfish and corals and so on. Hydrozoa, Cubozoa are cubomedusans, and the Scyphozoa, the jellyfish. These are put together in the group Cnidaria because they have a few things in common. One is most of them show alternation of generations, and we're going to talk about that quite a bit. They all have the same body plan. You can see the body plan on the second thing, upside down or right side up. They have a mouth and a stomach and little dangly things around it. And they all have stinging cells. So if you went to Angel Yanagihara's talk last week, you know more about those than I do, actually. But those are things that are typical of the group and characterize the group. And you're familiar with all these sorts of things. You can be a coral, you can be an anemone, you can be a cubomedusan, a jellyfish, Portuguese man of war are all different sorts of cnidarians. And this is going to be, the next slide is going to be a little complicated, but all I want you to know about is that it shows on your right side all the ones that have the medusae in their life history, the jellyfish, which is most of them, and then the ones on the left side, which are the anemones and the corals, which don't have a medusa. So we'll be talking about both of them, but this just shows you that in the group Cnidaria, most of them have this alternation of generations, and that's kind of our topic for tonight. Okay, all cnidarians show asexual reproduction, that is just budding off a little other one. So a little bud coming out here, a little bud coming out here. Corals often bud within the colony, okay, or a little bud coming off here. So you go from a polyp, sometimes you have a little planula, settles down and becomes a polyp, and this just goes over and over and over and over again. And it might go on forever. And that's kind of what we'll be talking about. Okay, the medusazoans, the ones that have the jellyfish stage, at some point, instead of going through this cycle over and over again, they'll produce a little tiny jellyfish, which will grow, turn over, and will become a male or a female. They'll reproduce sexually. So now the sperm and the egg, and now they have a planula, but this is a sexual product, so it has the genetics from both parents. Remember, this is asexual, and so it just is the same genetics going around and around. Okay, what makes this happen is different in different groups. The whole arrangement can be different for different sorts, but this is the basic life history kind of plan. Okay, so the point being that this stage could live forever. The jellyfish may die either because of reproduction and dying, or they may die simply because they're big and floppy and they get washed up on the beach. I was talking to Jerry Crow at the Waikiki Aquarium, and he said the Cuba medusans come in shore on a lunar cycle to spawn, and they die, but they mostly die by washing up on the beach. They don't die because sex was bad for them. At least we hope not. Okay, so the question for tonight is how to be immortal. Okay, so this is a big question. The best way to be immortal is don't die. But that's not much of a help. So a better way to look at it is don't get old. Okay, so there are some things you can do to avoid getting old. An important one I think we should all remember is don't become president. If you become president, in 8 years, you'll get old real fast. So that's something you might want to skip, if any of you are thinking of it. Okay, but what does it mean, don't get old? Well, we all know the stuff that goes along with it. Bone density, hair falls out, wrinkles come, muscle mass reduction, you lose your eyesight, you lose your hearing, and a lot of depressing stuff. But what's actually going on? What is getting old? What is getting senescent? Okay, all kinds of things are involved in it, and every kind of animal and plant gets old in its own way. But there's one thing that all living things, at least living things on Earth, have in common, and that is something to do with the mechanism of chromosome duplication. Something happens when cells divide and chromosomes duplicate, and this has something to do with, for everything that has cells in it, every kind of animal, every kind of plant can cause senescence. So we're going to talk about molecular biology for a while. Ready for, well, I can't even say it. Ready for molecular biology. Okay, every time a cell divides, the chromosomes have to divide. The DNA has to replicate itself and split and make paired chromosomes, and then the cell divides. So you had one cell here, all this stuff goes on, and you have two cells here. So DNA replication has to go on, or organisms can't grow. You can't make new cells, you can't repair, you can't grow, you can't reproduce, you can't do any of those things that being alive helps you to do. So this is how chromosomes duplicate themselves. I don't want you to memorize this. You memorize it if you want. The only thing I want you to see here is a part of a chromosome, and now here are two daughter chromosomes, and there's a bunch of stuff going on here, a bunch of complicated things, and all sorts of enzymes, proteins, and things hook on to the chromosome, hook onto the DNA strand in order to make this happen. So it's a really complicated thing. So all these things are hooking on, and doing the work of cell division. Okay, so you're asking yourself a question right now. Why is he telling me this? No, you're asking, these are all the things that these different molecules do. They unwind the DNA strands, they stabilize it, all this stuff goes on. But what happens at the end of chromosomes? Oops. At the end of chromosomes out here, when all this stuff doesn't have anything to hold on to. I need to make that thing go away, but I won't worry about it. Okay, thank you, whatever you did. So what happens at the end of chromosomes when this mechanism, these molecular machines don't have anything to grab onto and stabilize it? The answer is that the ends of chromosomes don't get duplicated very well because this machinery has nothing to hook onto. And so that means that any genetic information that's stored at the end of your chromosomes gets lost when the cells divide. That's not a good thing. Okay, the good news is that many animals and plants, including you and me, have a bunch of stuff at the end of the chromosomes that is sort of dummy DNA. It's DNA that doesn't code for anything, it doesn't make proteins, it doesn't have any genetic information. It's just a bunch of DNA out there in order to get wasted. If any of you have ever had a boat and you had to put zinc on the bottom of your boat to keep your propeller from disintegrating in the seawater, then you know what I'm talking about. Even if you haven't had a boat, I hope you know what I'm talking about. Okay, so here is a jolly young chromosome with lots of telomeres on the end. And here's a sad old chromosome, okay, with very little telomeres left, because every time the cell divides, those are lost. A few of them at the end just don't get replicated, don't get replicated, don't get replicated. Okay, and eventually you get down to the real serious DNA, the stuff that has the genetic information, and when that starts degrading, then you're in trouble. Okay, so when you run out of telomeres, what happens? You go into senescence. Okay, there's good news. Some special cells have an enzyme called telomerase that can add this dummy DNA onto the end of existing chromosomes. Otherwise, they can repair the loss that happened during the division. So how does it work? So now you're experts on molecular biology. You can see it's pretty obvious what happens is one of these things hooks on to the end of a chromosome and adds a bunch of just dummy DNA. In this picture, it's a sequence T, 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 G, 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 over and over and over and over and over again. And then when that chromosome divides, this will get worn down and more can get put on. Your telomere sequence and my telomere sequence is T, T, A, G, G, G. Those are the names of the bases that make up the DNA. So it's just dummy stuff that's on the end. Okay, as long as this telomerase is repairing the ends of the chromosomes, the telomeres remain intact, and so the organism will never senesce due to loss of genetic information in the DNA. I haven't heard any questions, so let's be great. Yes?
What percentage of the chromosomes is dummy stuff at the end, healthy chromosomes?
Okay, the question is what percent of the DNA in a chromosome is involved in the telomeres? A very small amount. In humans, the telomeres are about 2,500 base pairs. Okay, the rest of your chromosome is hundreds of thousands of base pairs, so it's a very small amount.
When you have a telomere for different people, what determines the dummy length?
You're born with a certain amount, and basically that's it. We'll get to that. So I kind of hinted at something, and I was really telling you a fib. Okay. For most normal cells in normal, what we call somatic tissue, that is not involved in reproduction, not making eggs and sperm and so on, the amount of telomeres you have when you're born is what you got. So this telomerase only acts in special cells in humans. So it acts in stem cells. Why, if you hear about people wanting to do research on stem cells, that's why it's so, people are so interested in them, because they have telomerase activity. So in humans, a number of telomeres, the amount of material you have when you're born is what you got. Unless you're making eggs and sperm, or eggs or sperm. You only get to choose one. Okay, so now I hope you remembered the jellyfish story, because otherwise you'd be wondering why I'm telling you all this stuff. Okay, if these little polyps that produce over and over and over again are really immortal, can really live forever, you would expect them to have telomerase activity, unlike you and me, who just have a certain amount when they're born. Okay, So a student of mine test for telomerase activity to ask the question, do these cnidarians have high telomerase activity? So my student, Ojiri-san, did this test for her PhD, and she studied 2 species of cnidarian. She studied the upside-down jellyfish, Cassiopeia, and a small solitary coral, Tinactis. And we'll talk about those in a second. Okay, this is Ojiri, and this is our host. Just so you know, it's a real person. They're both real people. Okay, so the upside-down jellyfish is interesting for this kind of study because the polyp stage does that asexual reproduction over and over and over again that I showed you in the beginning. Buds off these little things, they go make a new polyp. Buds off these little things, they go make a new polyp. Buds off these little, you get the picture. Okay, once they become infected with zooxanthellae, with the symbiotic, the same algae that's inside of corals and so on. Once they become infected, you can see these brown dots. They stop doing this, and they start making little medusae. And these medusae bud off, and then I showed you that silly little picture, and they end up being an adult. These are males or females, they produce eggs and sperm. So this is the life cycle I showed you before. With a little added twist that they have to have these algae in them to make medusae. Okay, Tinactis is, this is a coral from Okinawa. We don't have this here, but it's related to our fungia. And it's interesting because it's a solitary polyp, a single polyp. And it seems to grow up to a maximum size. And we think that also means a maximum age. Some corals might live for a very, very long time. You've probably seen pictures of giant coral colonies, but other corals seem to have a finite lifetime. They go to a certain size, a certain age, and then they die off. And this one seems to be one of those that seems to have an upper age limit. So you would expect older ones to have less telomerase activity than younger ones. Okay. So she developed 2 hypotheses, and they're just written down again. So Cassiopeia polyps would be expected, if they're immortal, would be expected to have high telomerase activity, and the medusae might not have that because they probably don't live very long, and maybe they aren't going to have that immortal life path. The same thing for the solitary coral. It seems to have a maximum age. And so you would expect that telomerase activity or telomere length would decrease with increasing size. So she did this. She measured telomerase activity in Cassiopeia, the upside-down jellyfish. She measured telomere length in the coral of different sizes and probably different ages. So what did she find? Ta-da, ta-da, ta-da. Okay, for Cassiopeia, what she found was there was quite high telomerase activity. That's good, but it was the same in all the stages. So she didn't find, the hypothesis wasn't supported because they all showed fairly high activity. So the hypothesis wasn't borne out. There's several reasons, and we'll talk about these at the end. Okay, what about the coral? Okay, the coral, on the bottom of this graph is size. So these are small corals and these are big corals. And here is the length of the telomeres. And you can see that as the corals get bigger, the telomeres get shorter. So this supports the hypothesis that as the animal gets older, its telomeres shorten, like I showed in that picture.
What does that C represent in front of the?
Let's see where.
In front of that.
This?
Yeah.
That's the name of the coral, Tinactis. It's a silent C.
It's a silence.
A silent C, like when there's no waves, right? No, I'm sorry. Anyway, okay. So what have we learned? about telomerase, yeah,
Previous graph,
yeah.
how many samples when you make that one?
Okay, not very many, because she had to kill the coral to do it. So both species had relatively high level of telomerase activity. Both species had relatively long telomeres. Cassiopeia didn't support the hypothesis. The coral did show decrease in telomere length with size, maybe age. Okay, so I said I'd talk about maybe why the Medusa, the jellyfish stage, had telomerase activity. And this kind of relates to the question of how many data points were in that graph. These animals all had to be killed for something else that she was doing. And so there's not a big sample size. And for the jellyfish study, the biggest jellyfish she looked at was about 10 centimeters. And if you see upside down jellyfish, you know, they'll get to be quite a bit bigger than that. So she may not have been sampling at the old age end of the jellyfish spectrum. The other thing is that, and this is something Maheilani knows about, jellyfish do have the power to regenerate. You can cut them and they will heal up and so on. So they do have the ability to heal themselves, to regenerate, and they may have telomerase activity that takes care of that. So that's still an open question. Okay, so that's what we learned about telomerase in cnidarians. What do we learn about the way science works? That science proceeds by testable hypotheses. So she had two hypotheses. The first one was polyps would have a lot of activity, Medusae wouldn't. And as often happens, it wasn't supported. And now there's some new questions. What would happen if you got really big Medusae? What would happen if you sampled after they did their sexual reproduction? These were just ones that she was able to find. So when your hypothesis isn't supported, you can go and have a drink, or you can make some new questions and say, well, that didn't work, maybe what is going on? Or you can do both. You can have a drink and make the new questions at the same time. And so this is the way that science actually works. You make a hypothesis, you test it. The same goes back to the question of how many points were in that graph. Obviously, you'd like to have more and more and more of them. That would be one way of improving it. The other way would be to look at some other anthozoans that we know only live to a certain age and have a look at them. Okay. No, don't look at that, don't look at that, don't look at that, don't look at that, there. Okay, so I'm ready for questions. And we're gonna all get kicked out at exactly 7:30, right? Okay, so we got time.
Yes.
When Maheilani asked me to do this, I said, I'm gonna give a 10-minute talk and leave 50 minutes for questions. And she said, well, we're paying you for more than 10 minutes. So I gave you more than 10 minutes of talk, but now we can have questions, discussions, or whatever. Yes?
I'd like to ask a question about relating to humans because of what you said on this Nigerian. I'm always wondering, humans live with different things, and the telomerase activity will determine how much telomere is on the end, right? How does one know how much is on one person and another person?
Okay, so the question is, how come you have more telomeres than I do? The question is, within a species, within, let's just say humans, since that's what we care about today, how do you know where you are along that spectrum? Well, there's a whole bunch of things going on. Okay, one of the things is, I said the average human embryonic chromosome has about 2,500 base pairs, averaged across people. Okay, as I said, each time there's a cell division, you lose some of those. and how much you lose is variable. It's not a fixed amount. So it turns out that that's really a lot. Most people in their lifetime, from being a single cell egg to being who you are now, haven't used up enough cell divisions to use up all those telomeres. So that is, a lot of people die of old age with still some stuff left on the end of their chromosomes, because there's other things you can die of. It's, we all know about that. Okay, so telomeres aren't the cause of death of most people. Okay, that is the lack of telomeres. Okay, but it is sets an upper limit on how long you can go on and have cells dividing. Now these guys are a little bit different than us, and for them, it's possible that running out of telomeres could actually have something to do with their life length. But for human, don't worry, don't go home now and freak out and try to get your, telomeres measured, because probably everybody in this room has enough to last them for a while. I can't guarantee it. But yeah. Any other? Come on, I saved all this time. Yes. Oh, yes. Yes.
So the Cassiopeia, that study was conducted in Japan, Okinawa.
Okinawa, yes.
So that variety is from Okinawa.
Yes.
Has she tried to compare it then to the Cassiopeia from other regions?
No.
She hasn't.
No. Yeah, that would start getting very expensive, and I think, to me, the more interesting question would be to start getting some really old, beat-up looking Cassiopeia, and see if, in fact, they are getting toward the end of their thing. Because a lot of animals and plants die of, well, think about, if any of you are gardeners, think about, if any of you are gardeners and live in Minnesota, think about what happens to your annual plants. Okay, they die, or die back in the wintertime, not because they run out of telomeres, because it just gets cold and they freeze, right? But the seeds or the bulbs or whatever, manage to keep going, yeah. So I don't know about differences among different strains of Cassiopeia. Turns out we don't even know what species we have here. I mean, we know one other species, and then there's some other ragtag things that showed up, so yeah. Yes?
What other kind of aging DNA causes a species to age? What other kind of degradation?
Okay, so the question is, what other kind of things makes us, well, this isn't exactly it. What other kind of things makes us get old?
In the DNA?
Yeah, I know, because I want to get to that. So, there are some, in complicated organisms like us, okay, there are different cells doing different things. And there's something, the only thing that I can think of that actually is death to a cell caused by DNA is what's called programmed cell death. Certain parts of certain parts of a body, okay, will have cells die in order to, well, for instance, if they're building neural networks, okay, you've got a bunch of neurons. Some of those neurons aren't going to function, and so those will die from what's called programmed cell death, and that's based in the DNA. But other than that, like the stuff that we're all suffering with, bad backs, weak knees, gray hair, wrinkles, deaf, blind, whatever, okay, those things aren't programmed in the DNA. Those are just things that, as the organism gets older, they happen. Now, what is philosophical thing? There's a question of why do we get old at all? And that's, it's a philosophical question first.
We couldn't count days, you would get older.
I don't know, you'd still get bad knees. You just wouldn't know how long it took to get them. Yeah. Yep.
Well, one of the big problems with damage to the DNA comes from environmental toxins, toxic rays, exposure to extreme forms of electromagnetic radiation. Those cause pinpoint mutations in the DNA, which may cause transcription errors in cell reproduction. And that also introduces a lot of problems with cell reproduction.
Okay, so
Over time, and with increased cell reproduction, those types of errors build up, and that leads to a lot of disease and old age. There's mechanisms in cells to check the DNA and try to make sure that it's been copied correctly, but sometimes those enzyme processes break down as well.
So the question is, what about external things damaging the DNA, whether it's toxins, radiation, whatever it is, and that certainly happens. And I had a bunch of slides in the beginning, which I didn't put up, having to do with corals, because this is really interesting to me. So I've swum around corals as big as this room. That coral is very, very old. It's in shallow water, gets a lot of UV radiation. It's been getting UV radiation for hundreds of years. And corals, we think probably most cnidarians, don't have a special cell line for making gametes. Otherwise, corals make eggs and sperm just out of their regular cells. So somatic cells, regular tissue cells, turn into eggs and sperm in these guys. That's not what we do. We have special cell lines that do that. So that means, you're right, these corals that are sitting in the UV radiation year after year after year are accumulating damage. Some of it gets fixed by these various kinds of repair things, but some of it doesn't. How can a coral do that? And what's interesting to me is if you have a coral that's 20 feet across, it's making gametes out of somatic cells, but a somatic cell over here had a mutation 100 years ago. A somatic cell over here had a different mutation 10 years ago. This coral is putting out gametes of different genetic composition. So we can't do that. Okay, the only genetics that you can put in eggs and sperm are what you got. But these corals are making mutant cells, essentially. And so they're eggs and sperm. And people are starting to get at that question. It's pretty hairy, and it's pretty interesting. Yeah. But yeah, so damage happens to DNA, and just to everything. I mean, my skin is getting damaged from the sun, so that's all kinds of external things. They just get old. The worst one is gravity. Yeah.
Okay, now that we have a study, there must be other studies. In your opinion, where do we go with this stuff now? Where do we give it more study, or do we do something?
Okay, so the question is, so what? Okay, so there's two ways of answering that question. One is, gee, we learned something about cnidarians that we didn't know before, and we have some other questions we could ask about cnidarians if we cared. Okay, but does that make our lives better? It made my life better. I thought it was cool, but it doesn't help. An average person isn't walking around going, man, I am so excited that those medusae had telomerase activity too. A lot of times when we ask scientific questions, we ask, we like to call it ta-dam-dam, basic science. Okay, and we just ask those questions because they're interesting and we can find something out. Is that going to lead to something wonderful? Well, Mahealani asked me to lead to something wonderful. Okay, this is kind of cheating. Okay, you can go online and buy jellyfish extract to make you live forever. Okay, there's some problems with that. Okay, one of the things is, so some people say, well, how can I improve my telomerase activity? Well, there's a bad way to improve it. Get cancer. Okay, cancer cells have very, very high telomerase activity. And so if you want a lot of telomerase activity, you know what to do. But you probably don't want to do that. Okay, the other thing you could do is get some telomerase, it's an enzyme, and take it. Okay, But the problem is, I showed you my little telomerase picture. Telomerase is an enzyme. Enzymes are proteins. When a protein gets to your stomach, what happens? It gets digested. So I'm going to ask a question, and don't hold up your hand. Does anybody, okay, if somebody here has diabetes or knows somebody that has diabetes or has a friend that has diabetes, you know that you can't take a pill, an insulin pill. Okay, you have to have an injection, because if you ate insulin, your stomach would digest it. Okay, they actually are working on an insulin oral dose, which goes in your intestine and attaches to the intestinal walls and does some crazy stuff. But basically, you can't just eat a protein and have that protein activity do something for you. So you can get this stuff if you want, but I wouldn't bother. Any other? Yes?
Family aging of these jellyfish, the aging plants. Look back, plants living forever. Because you take cuttings from one plant to another, you can speak to losing them. Turns out after many generations of that, we know there's no sex involved. There's no degradation of DNA. That if it's a fruit, it doesn't bear the same fruit.
Okay, so that had to do with organisms that we can basically cause asexual reproduction by taking a clipping of the plant and putting it in the ground and so on. Some plants take to that pretty well, and you get a lot of generations, some not so much. Which brings up an interesting coral question, which you were thinking of, and that is, if you swim around on the reef and you'll see a coral, and you look at it and you go, you know, this used to be attached, and you know, something fell on it, there's some dead parts in between, so now you have two pieces of the same coral. What's the age of those pieces? Is the age when that original coral was a single planula, a single polyp, or is the age when they got broken in half, did they start the clock again? And again, people are interested in that question. When these corals have this asexual reproduction, or either accidentally by breaking apart, or people doing transplants, you've probably all seen people breaking up corals and sticking them on little floats and trying to make coral reefs and stuff like that. Okay, and then your question comes up. So I got a coral, and I make 100 pieces out of that coral. I'm going to make 100 pieces out of each of those, and I got 100, and then, you know, I can repopulate a whole reef. But how old are those corals? And that's a really important question, and we don't know the answer. But yeah, for sure.
That the same question. You see this coral colony. Is it one animal or millions of animals? Because they're like homes in it.
Okay, so the question is, if you see a coral colony with hundreds of polyps on it, is that one animal or hundreds of animals? Well, luckily, scientists can make up words and say, well, it's a colonial animal, so don't worry about it. But yeah, it certainly works that way. Yes?
So last week we talked about the Medusa stage joining and becoming settling back on the surface and
Correct. Yep.
going back through it. I was just curious how that relates to that.
Okay, so when I showed you that initial life history slide, and then I said, sometimes it gets more complicated. Basically, it can get a lot more complicated. Okay, there are jellyfish that reproduce asexually out in the ocean. There are jellyfish, well, essentially they're jellyfish that actually attach to the bottom upside, not like the upside down jellyfish, but it's a different group of cnidarians. So if you've got a body plan that just has a bag and one hole in it and a bunch of arms, you can do pretty much anything you want. And cnidarian life histories are really crazy and complicated. But a really good one to think about is the Portuguese man o' war. Okay, so that is a colony, but the polyps do really different stuff. Some of them swallow things, some of them sting things, Some of them make a float to float around. So on a coral, most of the polyps kind of look alike, right? On a Portuguese man-o-war, the different polyps that have different jobs look completely different, but it's still a little floating polyp. So yeah, it could get way complicated. Yes?
Thank you, Dr. Bob, for coming tonight.
Oh, is it time to go?
Oh, no.
Oh, you sounded so formal. I said,
I have two questions. The first is, what cells were the telomerase activity measured from? And the second question would be, were the telomerase activity measured from a parent and an offspring, or were they just whatever?
okay, so the question is what cells were analyzed and was it a parent offspring situation? So the answer to the second one is easy, no. She just went to a pet store and bought some upside down jellyfish, right? Which is why they weren't very big. Okay, the other question is which cells? Basically, oh, let's go backwards. How come it's not going backwards? This is going to help you with your quiz, by the way. It's a little refreshing about a quiz. Okay, so she took cells, I have it up there? I had it, what did I say? She took cells from a polyp, a whole polyp. She took cells from a planulae for another one. She took a bell and she cut a piece out of the bell. She cut a tentacle out of the bell and she took a gonad out of the bell. But I don't think they were mature gonads. These have gonads, gonad primordia, you know. So yeah, so she had 6 different body parts. Yeah. Any other stuff? Okay, you ready for your quiz? Okay. Monitors, please pass around the quiz. Give people pencils. This is like, you know, when you're on an airplane and you're flying in and you have to fill out the agriculture thing and nobody has a pencil. And the cabin people go, well, we don't have pencils. You're supposed to have your own pencils. So nobody ever fills it out. And that's why we have all these introduced species. Okay, no more questions. Thank you all for coming.